I'm going to be talking about some of the work I've been doing in the past few years on the effects of spaceflight and mitigating these effects on the cardiovascular system. So we've explored space for decades, and humanity is finally approaching our goal of long-duration spaceflight. NASA is planning a trip to Mars, and there are even proposals for a permanent moon base, which would be very exciting. 1 second here. I'm going to get a laser pointer set up. Perfect. So while these plans are exciting, there are health effects for spaceflight that must be characterized fully and mitigated before we can truly achieve these goals. While the impacts of spaceflight on the body due to microgravity and radiation are diverse, acting on many different biological systems, due to both microgravity and radiation, we focused our study on the impacts on the cardiovascular system. In astronauts and in previous spaceflight studies, diminished cardiovascular function, occurrence of serious cardiac dysrhythmias, orthostatic intolerance, and impaired ability to respond to cardiac demands have all been identified as high priority risk areas by NASA. Previous studies have utilized animal models, and I really like the quote from your colleague earlier where mice are not humans. So the potential translation to human physiology is potentially limited. And the limited astronaut data that's been available has also been used as a big platform for these studies. To better study these impacts on the human heart, we wanted to develop a high-throughput engineered heart tissue model using human-induced pluripotent stem cell-derived cardiomyocytes, develop these engineered heart tissues, send them to the International Space Station, and study the impacts on cardiac function, structure, and the transcriptome. So our engineered heart tissue platform consists of a tissue suspended between two posts, one rigid and one flexible. When the tissue contracts, the flexible post is deflected, and we can use the Euler-Bernoulli equations to calculate the force of the contraction based on the magnitude of the deflection. This platform also allows for further contractile kinetic data acquisition, such as time to peak, time to relaxation, and the quantification of any arrhythmias. Here you can see, if you look closely, I'm not sure how visible they are over there, but you can see our tissues contracting in this platform. Unfortunately, like Christine mentioned earlier, the tissues are contracting, which means they're not fully mature, but we're hoping that our data can translate anyway. So astronaut time is also incredibly limited, so a goal in our platform development phase was to ensure that our platform enables measurement of contractile function in real time without the use of astronaut resources. To do this, we embedded a magnet in the tip of the flexible post and embedded these posts over a giant magnetoresistive sensor so that we can measure cardiac contractile function in real time without the use of imaging modalities. And space biology presents some unique challenges in terms of even just cell culture. Typical tissue culture vessels do not work in space, since the media would just float away in the absence of strong gravitational forces. So working with Bioserve Space Technologies, we developed a tissue chamber platform for culturing our engineered heart tissues. It has a hard polymer body and a gas-permeable PDMS membrane, and it's completely sealed, leak-proof, and self-contained so that the media does not escape on us. These tissue chambers are loaded into the plate habitats. 4 tissue chambers can be loaded per PCB with an embedded magnetoresistive sensor located underneath each tissue. And then a lid is placed over this PCB with vents to enable gas exchange and then shipped up to the International Space Station. And you can see these little ports here on the side. That's actually for needles for syringes for media exchanges. Next, we finally shipped our engineered heart tissues up to the International Space Station. On the left here, this noisy video is one I took on my cell phone during the launch. And here are some pictures of astronaut Jessica Mair on the International Space Station performing a media swap. Sadly, these pictures were not taken on my cell phone because they get really mad at you when you try to stow away on the rocket. So while in space, the tissues were maintained viably for 28 days before returning to Earth. During this time, a subset of tissues were stored in paraformaldehyde for structural analysis or RNA later at different time points for RNA sequencing analysis, and this analysis is still ongoing. However, we did indeed see cardiac tissue dysfunction in space. This, I think, is one of those very rare occurrences in life where I can say I was excited to see cardiovascular dysfunction. During spaceflight, the twitch forces decreased significantly over time, while the mean change in beat interval, a metric of arrhythmia, increased significantly during the majority of the stay on the International Space Station, but returned to baseline upon return to normal gravity. We also found significant decreases in the upstroke velocity that do not return to baseline upon return to normal gravity. And the time to 90% relaxation also slows. This data was intriguing as prevailing theory indicates that reduced afterload should accelerate myocardial relaxation rate, so the impacts here could be independent of load. However, mitochondrial dysfunction and subsequent metabolic changes could also lead to an inability to meet the energy demands for ion channel activation for both the relaxation and contraction phases. Mitochondrial dysfunction could also lead to the decreased twitch forces and the spaceflight-induced arrhythmias, and has been identified in previous studies on the impacts of spaceflight. So as I've very so subtly hinted at, our prevailing theory was that mitochondrial dysfunction could lead to this cardiac dysfunction that we've identified. And indeed, in our late spaceflight samples, these are day 28 spaceflight samples, we do see mitochondrial ultrastructural defects in spaceflight. So first you can see pathological levels of lipid accumulations next to our sarcomeres here. In our next images, you can see rampant mitophagy and mitochondrial ultrastructural defects such as paracrystalline inclusions, concentric laminated bodies, and compartmentalization in our mitochondria. And I think this mitochondria here sums it up quite nicely. These are very sad, frowny face mitochondria. Next, after characterizing the impacts of spaceflight on the heart, we wanted to initiate preliminary Earth-based drug screens. To enable this, we utilized a random positioning machine, which is pictured here. Shown in this picture, the PHAB is placed on top of this random positioning machine, and it rotates randomly, as the name implies, to simulate microgravity by essentially forcing the gravitational force vectors to sum to zero. So we compared our control tissues cultured off the RPM, which you can see in black here, to tissues cultured on the RPM with a drug that we unfortunately do not want to disclose at this time, as well as just control tissues that were cultured off the RPM. And you can see that there are statistically significant decreases in twitch force at week 3 on the RPM cultured with no drug that we do not see in the RPM cultured with drug or in our control tissues. And interestingly, we also see, so this is a baseline recording before we put them on the RPM of one of our RPM tissues, and at week 3 we saw our first tissue on the RPM that exhibited an early after depolarization, a type of arrhythmia. We did not see any of these arrhythmias in any of the tissues in the RPM drug treated or the control tissues. And by week 4, every single one of the tissues cultured on the RPM had arrhythmias, and again, they were not present in our other tissues. So while we continue this preliminary drug screening, we're very excited to launch our engineered heart tissues to the International Space Station again this October, where we hope to identify one or more mitigation strategies that can prevent the effects of microgravity on the human heart. So the work that I've discussed so far has mainly focused on one of the two main perturbations of spaceflight, mainly microgravity. However, there is another major risk factor in space, that is galactic cosmic radiation. Galactic cosmic radiation consists of both the proton and alpha particle heavy solar radiation, as well as the particularly damaging high charge and energy transfer atomic nuclei traveling at near relativistic speeds thought to be generated by supernovas. These ion species are difficult to shield against and remain one of the biggest health risks for astronauts in spaceflight outside Earth's magnetosphere. These risks are evident when you look at the proportional cardiovascular mortality rate of Apollo lunar astronauts compared to non-flight astronauts. So astronauts are in peak physical conditions, so they typically will have lower cardiovascular health risks compared to the normal population. But when you compare within the astronaut population, the Apollo astronauts, the only members of our species that travel outside of the Earth's magnetosphere up to the moon, have a significantly higher proportional cardiac mortality rate than non-flying or low Earth orbit astronauts. So clearly the impacts of space radiation on the human heart can be devastating. And previous studies have relied largely on monoenergetic single ion beams, which do not accurately recapitulate the full damage caused by these multi-ion beams. They've also relied largely on animal models, which Chris Hughes would be a terrible reviewer to try to pass those through. So while we've learned a lot from these previous studies, we set out to utilize our engineered heart tissue platform to study the effects of simulated galactic cosmic radiation on the human heart at the NASA Space Radiation Laboratory at Brookhaven National Laboratory. So here you can see our heart tissues up at the top shelf here in front of the accelerator. Here's the very end of the accelerator, and we transported our tissues there, and as you'll see in this video, we irradiated them. So while still not actual galactic cosmic radiation, simulated galactic cosmic radiation at NSRL consists of both the alpha particles and protons present in solar radiation, as well as several species of the particularly damaging high charge and energy ions to more closely mimic space radiation. We utilized 3 doses to establish a dose-response curve. These doses consisted of 10, 25, and 50 centigrade. And while a lot of this data is still quite preliminary, I'm very excited to share these findings with you as long as we keep it in this room, because I do not want to restart my PhD if you scoop this from me. So first we identified increased ROS presence in a dose-dependent manner after 24 hours using the CellRox dye. Interestingly, this trend is maintained at day 30. However, it's not statistically significant due to the large range of noise that we found. Looking at the interval irregularity, again, that we've discussed earlier in this talk, just after 24 hours following exposure to the beam, we do have a statistically significant increase in this interval irregularity that is further exacerbated as time goes on. And then we also took a look at the mitochondria using MitoTracker green and red. So MitoTracker green, canonically, there is a little bit, I know, controversy about whether or not this is true, but it should stain all mitochondria completely independent of the membrane potential, whereas MitoTracker red, CMX-ROS, is sensitive to the membrane potential. So using these dyes, we looked at the mitochondria, and without even quantifying any fluorescence intensity at this point, we did notice following 1 week, we did have a significant amount of mitochondrial fragmentation that we're currently analyzing further. So this correlates with previous work that has identified oxidative stress and mitochondrial dysfunction as one of the central hubs for spaceflight-induced tissue damage. So we're excited that our data is recapitulating a lot of the previous studies. We next quantified cell viability using the dead cell stain Ethidian homodimer 1. We did not see any significant increases in cell death until day 30, which we thought was really intriguing. At day 30, we did find a dose-dependent, statistically significant increase between 10 centigrade and 50 centigrade increase in cell death. We hypothesized that this delay in cell death until 1 month following exposure was due to the compensatory mechanisms for mitochondrial dysfunction and oxidative stress being eventually overwhelmed at these later time points, leading to this cell death. And we're continuing to look at this in our current studies. We also stained for DAPI to label the nuclei and gamma H2AX, marker for double-stranded DNA breaks. And we did find double-stranded DNA breaks were prevalent in even just our 25 centigrade 30-day time point. And so this indicates that DNA damage is present in our tissues. And this DNA damage can result, again, both directly from the radiation or from the oxidative stress that we've found. We're continuing to characterize these effects, and we're hoping to finish up this characterization actually within the coming months and start screening our mitigation strategies in the coming year. So with that, I'd like to thank the funding sources, including the NIH and NASA, the Kim Lab, and especially my PI, Dako Kim, for his support, Lila Stoddard, who's been absolutely integral in this work, and my undergraduate, Jeff Chen, who's going to be applying for PhD programs this fall, so keep an eye out for that application if you'd like a fantastic applicant. We also have many different collaborators, such as Bioserve Space Technologies, Sharon Garrett down at Duke, and special thanks to the team at NSRO.